Hi, I'm presenting today a study from 2012 that conducted phase 1 clinical trials to test the safety of the intracoronary infusion of Cardiosphere-Derived cells, and also assessing whether or not the treatment delivered the intended outcome. Next slide. To put things in perspective, a myocardial infarction leaves scarring of the myocardial tissue, which was believed to be an irreversible change. The cardiomyocytes do not regenerate, leading to poor disease prognosis post-MI. So previous studies were done with bone marrow mesenchymal stem cells, but the outcomes were inconsistent. So here, in this study, Cardiosphere cells that are aUtologous to the patient are therefore present, which would therefore present no risk of immune rejection. So this study was randomized, conducted at 2 different sites in USA, 2 different hospitals, including Johns Hopkins. The participants were divided into 2 groups: CDC treatment group and control group, who received standard of care in a ratio of 2:1, 17 treatment to 8 control patients. The CDC cells were infused through intracoronary artery with prior stent in place. The results show not only regional improvement in scarring, but also increase in viable myocardium. And this study was concluded to be a successful phase 1 safety trial. So I'll briefly explain over here the trial, the study plan. 336 patients were assessed for eligibility and ended up with 23 randomly allocated CDC treatment group and 8 random. 6 had to be dropped off out of the 23 due to different reasons, as listed in this picture below. And then 17 received CDC treatment. The first 4 patients received low dose of 12.5 million cells, and safety review was done, assessed by the safety monitoring board, and allowed them to go on to a higher dose of 25 million cells. There was 1 patient who received intermediate dose of 17.3 million cells. This patient was enrolled on compassionate basis because they were 14 months post-MI. There was initial screening and MRI blood tests done, and then done at 6 months and 12 months. These were the primary safety endpoints as well, listed briefly as sudden death, tachycardia, myocardial infarction after cell infusion, tumor. And the secondary safety endpoints would be hospitalization, increased cardiac enzymes, or renal hepatic abnormalities. All right. Okay, so the explant culture is grown from patients' heart biopsies. So the heart biopsies were taken, grown on the plate cultures, and they made these cardiospheres, as shown in Figure A. These were then transferred to the suspension cultures. So the figure E in here, so you can see B, C, and D, they show the photographs of the cells growing, and by the time we've got D, there's already they're starting to connect together to make a network. The figure E shows how many passages were needed in each group over the number of days to grow either the 12.5 or the 25 million cell dose. Is depicted by the red and blue colors there. The identification of the cells was verified by looking at the CD105 and CD45 markers, and they had CD105 had to be more than 95% expressed, and CD45 to be 5%. So this was verified to be the correct cells. So here I show a video of how these cells in suspension would look like. This is not from the study, this is just taken from YouTube, just something interesting to look at. So just as a diagram of how the procedure was done, which was already explained: biopsies taken, cultured, and inserted into the affected region. Slide. So here, this is one of the major results that are shown in the study: MRI of the treatment of control groups at baseline at 6 months. So the CDC group on the top, control group on the bottom, and on the left is the baseline, and right is the 6 months result. The lighter area shown by the arrow is the scarring, and the darker areas are where the scarring has healed. So you can see in figure B, at 6 months CDC, there's more darker areas as compared to D. The graph also shows that the scarring had reduced in the CDC group, depicted by the red histogram bars. So there was a supplementary study done in rats to show that the myocardial regeneration is independent of the MRI results. So the heart after the ischemia was given to 45 rats, and the hearts were biopsied, stained, and the blue area in the control group shows more scarring than in B. So the follow-up studies were done after the March 2012 original study. July 2012 is a correspondence, which they're talking about which fraction of the CDC cells, whether the cardiomyocytes or vascular endothelial, were involved in this regeneration, and are CDCs of a patient different from a healthy CDC. In March 2013, these results of this paper, which is also a follow-up paper of the same group, they're showing 45% reduction in scarring. However, there's not a lot of supplementary data in these results in this publication. In 2014, the follow-up phase trial, it's showing the full 1-year follow-up. They have a bit more detail into follow-up testing, comparison and verification tests, and statistical analysis. Since then, I wasn't able to find any other ones apart from some of these studies, which have taken sort of a different direction into allogenic Cardiosphere-Derived cells, like this one is in 2014. And it's about a CDC infused into a swine heart. Another one that shows it's a preclinical study of allogenic CDC in immunologically mismatched animals without immunosuppression. It was found to be safe. So they use mesenchymal cells. To summarize, the limitations as stated in allogenic studies is that the autologous CDC approach is more time-consuming, expensive, and requires specialist facilities. The major limitation of this study, however, is that there is no significant improvement in left ventricular ejection fraction. However, it did show good regeneration of viable cardiomyocytes in the affected region with visibly reduced scarring. This just shows that the therapeutic regeneration is possible in cardiac tissue. Future studies need to be done to show how are CDC impacting regeneration in the affected region at the molecular level. And is there going to be a phase 2 trial? Thank you.